So, good evening, everyone. I hope you have received the papers which you have asked me yesterday regarding Jivas. You all can go through it and get from the basic to the high end understanding of all the aspects of Jivas. So, now we will be starting from the point we stopped yesterday.
So, ma'am, you can start now. It's already 6. Hello, Priyanka, ma'am. You can start now.
Yes, I'm starting. I'm sharing my screen. So, yesterday read we in our analysis we have identified we have got the SAM file, that is the sequence alignment map format file, that after aligning our reads to the reference genome. So the next step in is that we have to, because SAM is such a format, which is such a heavy size format which cannot be used for further processing. So we have to convert that SAM format to BAM format, which is a binary alignment map. It is just the conversion of this SAM file to the BAM file. I would like to explain you that what are the headers in the uh, BAM file, means what how BAM file is different from SAM file. So you can see the new screen, right? Yeah. So if you see in the BAM file, the BAM file is has the header sections. What headers are there in that header? First header that contains a sample name, its length, alignment method, alignment, and details about the alignment, that the read sequence, the read quality, alignment information, custom tag, which includes the read name, includes the chromosome name, the start coordinate of the alignment quality, and the match descriptors. Then the alignment section, this alignment section also include these information in this in our BAM file, that is RG, which says read group, which indicates the number of reads for a specific sample. BC is mentioned in it, which shows the barcode tag, which indicates the demultiplex sample ID. SM is the single end alignment quality. AS is the paired end alignment quality. NM is the edit distance tag, and XM is the amplicon name tag. These are the detail of the alignment file, what headers it has, the BAM file. So now how to convert the our BAM file to the SAM file. Okay. So you can see my slide. Everyone can see my slide.
Yes, ma'am.
Okay. So in the slide, I have shown you that for converting SAM to BAM, we have to use SAM tools. So if you do not have SAM tool installed on your systems, then kindly install SAM tool. I will show you, I will give the in the link the path from where you can get SAM tools. My screen is visible. The size of the screen is visible to you.
Yes, ma'am. Terminal is visible.
Okay. So I am installing SAM tools one other way, sudo apt-get install SAM tools. Sudo apt-get is to be, so it will install SAM tools with in the Ubuntu OS. I already have, so it says that it is following package was automatically installed and is no longer required. Or if you have problem in installing by here, you can directly go to the SAMtool page, download the source version from here. I will copy this page. This both the options I will tell you in your chat. This is the link of SAMtools where you can download SAMtools. Okay, and if you have a Linux operating system, you can just write. Okay, so this way you will get, this tool will help us to convert the SAM to BAM. So as we have several BAM files, we'll convert all of, because I have already converted and send you, like this is the SAM1 sample 167. This will be can be converted, will be converted to SAM BAM, to BAM 167. How to do this? This is there in the command, is in the, here I am showing SAMtool. You have to write SAMtools view dash s dash b, then the input SAM file, and then the output BAM file. I will copy this command in your terminal in the chat box. Convert for conversion. This command, I will show you how it works. So I have typed, because I have the both my files in the same folder. So I will give my output in a new folder. So this is my SAM file, which is input, which is present here. I've given the option SAMtool view s dash s dash b, sample1 SAM and sample1 BAM. Okay, it will get converted. It will take some time because the file size is not so small. Okay, so till that time I will explain you the rest part. So see, once our BAM got prepared, like this we will prepare BAM for all our samples. See, as I told you that for, and the papers also I have sent you, you could see very nicely that this GWAS analysis is not performed on a smaller number of sample. It is always performed on a very large number of samples. So I have shown you conversion for one sample, and I have given you as a test for 4 samples. You have to convert all 4 samples, but the actual sample, this is one sample, that is sample 1, but the actual sample will be much more than this one. So now I will be when, because we will do all the analysis with this 4 set of sample. So I am showing you one, and rest you convert it. Okay, and then now next step, once we get our BAM format, alignment format files, now we have to identify the variants. For that we have to use it. There are many tools which can be used for identification. Here I'm explaining you Stack is a very popular tool which is designed as a modular pipeline to efficiently curate and assemble large number of short read sequences from multiple samples. It supports all the major, and the good point of this is that it supports all major restriction enzyme digestion protocols such as RADSEC data. Also it can use, we use double digest RADSEC also, and a subset of genotyping by sequencing protocols. These type of data can also be run using the Stack. I'll give share with you the Stack link also. You can read on the main page of Stack. This is Stack. Just type Stack. When you type stack, you will get this link. This is the manual of accessing. So So now I'll come to explain what it does, or this stack, and what it does, it identifies the loci in a set of individuals, either by de novo or alignment to a reference genome, and then genotype each locus. The stack incorporates a maximum likelihood statistics model to identify the sequence polymorphism, because here our aim is that we want to find the SNPs and check how they are in the population, means how polymorphic they are in our population. Stack employs a catalog of to record all loci identified in a population and matches individual to that catalog to determine which haplotype allele are present at every locus in each individual. Actually, it is explaining its algorithm that it employs a catalog. There is a catalog based method in which it records all the loci which it has identified in the population. Like we are using a few four sample, the complete population is of more than 100 samples for our individuals. So all will be used and then match the individual to that catalog, means after identifying the individuals in every sample, it will match to the catalog is a record which is completes the detail of all the individuals, and then it will help to determine which haplotype allele. Haplotype is basically is a continuous stretch of SNP in a region when there are several SNPs in a present in a stretch of loci. Then that region together, and it is getting forwarded in generations together, then we call it a haplotype. So we will be able to, and this will determine the haplotype allele which is present at at every locus in an individual, because it is combining the data from all the individuals. And it can handle both single and paired end Illumina sequencing and compressed raw data also. So can anyone tell, have you got the BAM file? I have got the BAM file. My command is complete. Okay, I hope you must have got. So the next, we will be starting with the next step. The next step, what are the major steps in the stack that I'll be explaining you. Just I'll take a 1 minute break.
Most let's see. Just let question.
Yes, I'm back. So I was trying to explain you that how does stack works. What are the major steps in stack? First, it clean and truncate the data. Stack reads. How does it does? It reads stacks. The read have to be truncated at a particular length, discarding those reads which are below a length. Means this is talking about the input read. These are the different step in the stack program, and it is telling about the input raw reads FASTQ. How does it process? So it discards those reads which are below the specific chosen length. And then if have a reference genome, if you have a reference genome, then reference map.pl script is used to align the data against the reference genome. If we do not have a reference genome, then de novo map.pl is used. So we will be using both these pipelines uh, to run our to identify the variants, both ref map PL, de novo. And as it is obvious, the reference based mapping is always faster because it has a already staged reference genome, and then we map our reads against it. But for a de novo mapping, it's always a bit more time taking, as we do not have a reference genome. So what steps it follows, this is the internal steps which because to us, if we can run separately these steps also, it gives the option that you can run u stack, c stack, s stack, all these separately, or but there is a script, this ref map pl and de novo map pl have integrated all these steps together. So what does u stack do? It basically is an assembly loci in each individual and building stack. It makes an assembly of loci of each individual, and by individual stack it means a region. Then C stack build a catalog. Catalog is the information for by catalog is formed by combining the U stack information. S stack, what does it do? It matches the parent and progeny to all generic samples against the catalog. It compares your parent and based on what your data is, either it is parent or progeny, or it is a, or you are talking about high low data set. Then what it does, that step is TSV to BAM. So here it transposes the data from being stored per sample to stored per locus. Then means the output we will be getting at this step, we will see that we'll get the per locus change. And then the GStack assembly to assemble paired and contigs and recall the SNP using population wide data. Means it considers the complete data set for the paired and contig and it recalls the SNP again. And population step map, which determines which group to use for calculating summary stats, means which group, because here it is a very uh, big advantage of this software that it takes the information about your population also, means what type of data, either you are dealing with some high low samples, or you are dealing with parent progeny sample, or you are dealing with what is your sample type. According that information it incorporates during the run. So if you see the broader picture of it, this is the broader steps which we have discussed in detail. That is, it processes the rad tags is the reads, it add the raw reads in which it adds the tag. Then the u stack, p stack, c stack, s stacks, all these steps which we have discussed. Then it calls the genotype, that is in individuals, like here it is male and female, what are the alleles present. Then in the population, compare this with the complete population. How is the trend in population A, population B? Because you can give more than one population in this. Okay. And then the output it gives can be used for the it gives in the form of the file. It can be used, it can generate some, give you a VCF file, genpop file, filef file, all these type of files it gives as the output. Okay. So now I will be running stack. I feel you must have got BAM file. Few files I have already given you. Uh, yeah, who has shared this? Yeah, this is refmap PL and this Dino, right? So now the next step is that we will be running refmap PL. Okay. So for installing refmap PL, what you have to do, you have to go to stack X first, stack main page. You will get download stack. Once you will download it, you will you can see the manual. It will tell you what are the steps. Basically you just have to extract the file, then run the make command, and then configure it and run the make command. These all steps you have to do. I will copy this in your chat. Yeah, so these step for the installation of the stack, and here you will change according to the version you have. Okay. So once it is installed, if you all install it, and you all have the BAM file converted with it. What else is needed? I will explain you. Run refmap.pl. See, this is, they have explained that the two pipelines, both there is one refmap PL and one is de novo map PL. Refmap PL is used for reference based mapping for identification of the variants. So first I will be explaining you what are the inputs required for refmap PL. See, this is the biggest one advantage of using stack is that it will take information of your population. So you have to give the details, the population map. That is, population map is what is a text file. We have to give a text file as input. I will show you how to prepare it, which will contain two columns. The two columns will be the prefix of each sample in the analysis. The two column will have, the first will contain the name of the sample, and the second column will be what type of population it has. Means like I will explain you. For example, see, like here this is a population. Here there are fish. Here a more realistic example how might analyze some data with more than one population map. For example, if it consists of the individuals which have red, the fish in the red lake, the blue lake, the orange lake, and the yellow lake. This is a double combination. First I will explain you simpler one. Like, see, simple population map is that it has 6 individuals and 2 population, in which means it is saying that the integer indicates the population can be a unique number. It doesn't have to be sequential. This number can be a population number. If you want to say individual of sixth population, individual two is of two is also of sixth population, this can be any number. It is not that you have to take a specific number. Like, for example, you can write it at as that these two individuals are from of FW population, and these individuals are of OC population. Okay. Like other example is like for this population has two types of changes. Like it is in red lake, blue lake, yellow lake, orange lake. So we will these individual 1 2 3 4, we will write 1 2 3 4 red as the location. They are specified to be from red lake population. Then 5 6 5 6 7 8 are from the yellow population. And then 9 10 11 12 are from the blue population, and 13. This map we have to prepare population map for our file also. I will show you how I have prepared for my file. Like this is another example. Like it has high plate is these all are high plated and these all are low plated. So how we will write these all individual, we will write one sample 1 to sample 8. We will write high in front of it, means these are belonging to high population. Then and because 9 to rest 16 are low population, so we are writing it low. Then so now if we want to combine the two, we have such a population which we have both the information, also the phenotypic information, that what type of lake they means phenotypic means either they are high plated or low plated, or we also have the geographical location, that is they are in the red lake or they are in blue lake. So finally the population, how can we prepare population map for this type of population in which we have more information? We have to mention 3 rows, 3 columns in this. One will contain the type of which was in the first above example I showed you. Will for first column will contain the that which are belonging to red, which are yellow, blue, and the additional that either they are in high group or they are in low group. So an additional column will be added. Okay. So as here we are using genetic population specifying a genetic mapping cross. So he the population today we are using is also a genetic mapping cross in which we have parent and parent samples. Parent are always 1 and 2, and the progeny is obtained after the cross between 1 and 2. So this is the parent, and these are the progenies. So it is mentioned progeny. So how will because I have just done the we are doing the analysis with 4 sample, as we cannot make it more than 4, it will not be feasible to run. So I have given this information. I have made I have opened a text file. I'll show you how I have made. See, for the reference based run, I have made a population map file. I will show you that file. Pop map. This is the pop map file. I named it pop map. I have given sample name. Like sample name is our 4 samples we are using: sample 1, sample 7, sample 168, and 167. So these 168 and 167 are progeny, and 1 and 7 are parent. So the progeny must be very big. Parents are always 2, mainly in a bicross. So this 2 actual SAM file will contain several samples in the progeny, more than 100 or more than 200 or more than thousands, based on what your experiment is. So I have, according to our data set, this is the file which we have prepared for the population map. So the next step comes. Now we have to, before running this ref map pl, see, I feel all of you must have installed ref map pl. I should proceed further. Should I start running ref map pl? Okay. Okay.
Yes,
So
ma'am.
yeah, ref map pl now once installed, I'll show you. Now you go to the stack directory. You type ref map pl in on your terminal. As you can see my terminal. So once you see that, what is it does this ref map, because this is reference based call identification of variant, as I have explained you here on my slide, that we are identifying reference based variant using ref map pl. So ref map pl, what it needs the input output sample files, the and which file it needs the alignment file, that is the BAM file or the SAM alignment. We have converted our SAM to BAM, so we can directly use it. What else it needs? It needs a pop map file, that is a population file, path to a population map file. Then what does it need? It needs the spacer for file name, that is the empty. And this is the empty, and program looks for the file. Basically it is a spacer. By it means that if it is named, what is the name of your file? If this option, for if you are giving the name sample 1 name 1 sample, the name will be how it will be named: sample name 1 spacer, there is a space, and then the BAM. So it is just the sample name you have to give it. You don't be worried about the spacer. So, and the other important thing which we have to give is that after that, the population map, which I have told you how I have to we have prepared for our files. Then the out path, where you want your output path. And if you want to remove the PCR duplicates, you have to write r PCR duplicates, this option also. And then other than that, whatever files you want to generate, if you want to generate a VCF file, then you have to give that option to it, then it will generate all the files. So I will be running this command to you. I've already run once, but I will again run. Yeah, see, I have run. I will show you. Yes, ref map pl, what I will copy to you. These are the minimum things which we will type. We can add many more outputs to this command. It is not so that you just have to type these many things. If you want additional file for your file lib, if you want some other files, this also you can get. It won't be an issue. So So actually here what happened, that no such file is, it is saying, because we are must be not be in the right directory. Okay, so no such file popmap.input. Okay, so now what we'll do, that's what I have given. First I will explain you. It is mentioned that we have to give the TT is the thread, pop map is the population input, which I showed you the file which I have prepared. I will again show you once. Pop map, this is the pop map. Okay, which contains the parent and progeny name. Okay, and the sample file name should be exactly what you are using. Like here I have named it as I will show you what I have named. Uh, yeah, I have named it pop map, the sample 1, sample 7, and sample 167, 168. I've given you that samples only, so you have to exactly use this only. You don't have to change the name. Okay, and don't leave any, there should be a tab in between this, then only this file will be accepted by your software. Okay, so now we have given the output path to the stack reference based output. This file folder I have prepared already, and the samples are present in our training data set. That is, here are the samples. I will show you. I've kept them separately, the BAM file. These files are present here. So these all files are here. This can be used by our program. So, and here also from wherever you are running, you should give the right path so that it can get the BAM file. So I'll be running it from here. It is saying, okay, we will not give this option. It is not taking, this version is not taking. We will remove the PCR duplicates. Okay, so we will using the option RM dash PCR dash duplicates. Okay, still there is something unknown, option 9. Okay, yeah, the 9 got typed. Now what is there, that we have given it's the path to the sorted. We should give it path here. It will run. Okay, so actually the file popmap file which we have prepared, that should be very carefully prepared. So it is showing that error only. That only I said that popmap file should be tab delimited, because when I have run earlier in the day, I'll show you what was the result. It was, I have changed it to tab delimited, then it was giving the right results. So I'll show you that part. Yeah, see here it has run. So it's a refmap plt popmap input. This file I have tab delimited this file. So then finally, after doing the tab delimitation of the file, input was my reference based input, output path was stack reference based output, and samples were in this folder sorted BAM, and the name exactly should be given. So I found all the what output it gives. I will explain you the output. It called the variants, genotype, and haplotypes, all these, and I have the output files. And finally refmap pl was done. Once it is done, it will give you this. Okay, so everyone has got this message. I feel you must have got.
Please, Mark, can you Sorry, ma.
Yeah, please say.
Can you resend the edited
What? I didn't get you.
the command line you used, the edited one?
Edited one. Okay.
Yeah.
Actually, you have to prepare that pop map input file that that is prepared by myself. It is just a text file. I've explained you how to prepare it. Someone is writing yes, so I feel they have prepared the right output file and they got the results. Okay, and if you are not able to prepare the file, then let me know. I will explain you again. So once it is ref map PL is complete, the output which we will be getting, I will explain you about that. See what outputs I got. The output contained the main final, which is the important output, is the population sum summary stat, which will help us to explain what actually we got, how the variance we got. So I will open it. So this file shows the summary of the variations, the standard deviation, overall summary of the what was the because our see what is that data set what we have taken is very small. The actual data set of this is, I will tell you, of 300 samples, because it is not possible to run all the 300 samples here, so we can only do with this number of samples. So if you see the results, how much these were the this was the parent sample, this was the progeny sample, how many sites were there, you can see it shows that there were 5,89,835 sites in parent and 4,93,301 site, in which the variant sites were 22 here and more variants were in progeny. And the polymorphic, that is the site which shows change within the within the within the population, that is at a single position there is change of the nucleotide within the population, that was 22. All 22 were polymorphic, and in progeny also all 22 were polymorphic. Then percentage, if we calculate with the total loci, this was the total number of sites and these were the percentage of polymorphic. So it is very less as the population size is very small. These are all values, statistical values, what was the variance, standard deviation, p value. It is based on the your input of a number of variants you got and in how many individuals. One is this summary file. Other file which you will get as output is, I'll show you that file. The other file you will get, which will contain, which will show you at which location, this is the chromosome number, this is the position, at which position your locus is present. Okay, so I will in the progeny and your parent, I will explain you this in more detail. With this is very clearly mentioned how is this file prepared and what information this contain in the stack tutorial. I will explain you from there, because they have very colorfully explained it, so it will be easy for you to understand. See how what outputs we get. Uh, that output also I will say we got the population summary stats which we have discussed. It's a population sum stat TVS, which is the summary stats of each population. Then we got the population sum stats, this is that only more detail. Then we have got this file also which calculates the FST. FST is calculated for each pair of population and for each variable site between the pair of population, and a p-value indicating which indicates the statistical significance of the difference in allele frequencies. And the file which we are discussing, I will open that. First we will discuss that. Yeah, it is this. See, now we got this file which we are discussing, that is locus summary, which is giving the population stats per locus. So how that data, this data file which we were obtained, what does this represent? I will explain that to you. See, we had a reference based assembly. This is our reference from 5 dash to 3 dash, from 4085 to 4120. This is the region which we are just explaining for an example. These are our sample for the locus 1 and there, and for the locus 2, the two locus we are comparing here. So if you see that this was the reference, and here there was a restriction enzyme site. So our fragments which we obtained from sequencing run are present at this region only, because there was a cut in our input sample from here. So if you see that in the locus 1, what this is the blue part which is shown here, how this is represented in our output file. It says that in the locus 1 at position 4108 position, that this position is 4108, what it found that it has A and G. These alleles are present at this position. And at position 4116, it finds that it has G and T allele present at this position. And if you see this, for the second locus at 4095 is this position where the alleles find in our population was C and T. At the position 4092, we see that the allele are T and A, and at the position 4086, the alleles are A and C. Either the A slash C is show present. This result is represented in our Excel file, which we have obtained as a result in our parent progeny data. So what other files which we get? We can get the VCF file also. VCF file is very population. It is a very popular file. It is a variant call format file. It is much easier to understand. See, it says that we have a chromosome, we have a position on the chromosome. Then means we don't give, the software gives when it generates VCF, and how it is showing the reference based calling of the variants. This was A was present at the reference, and what was the alternate allele we got was G. While in the 4116 position, G was present at the reference, and what we got in our sample was T, the alternate allele. Here it says that G was present in this, here the change, the G was present at this position G, this, it is talking about this position. In the reference there is G here, but in our case majority of the alternate alleles were T. Okay, then sorry, I'm so sorry, GA, majority of alleles were A. Then at reference A position, it was T was found, and at reference T position, what was found? G was found. So the VCF file, I will explain you about more about VCF file. Actually, VCF is a variant call format file. It is it has the header, what information it has. It is very commonly obtained file after a variant calling from majority of the tools which are used for identification of variants, the SNPs. What does it contain? First, it contains the chromosomal information, the position information, the ID if identified during the call, the reference, what was present at the reference allele, what was in the alternate is the means in our samples what was there compared to the reference genome. And this is the quality, what was the quality of the call, whether it passed the filter or not. And then if there is annotation information, if you have given it, then it will show you. Then it shows these four important information which for all the sample, like these Z115 ZW177 are the samples. So it says that GT AD, GT represent the genotype, what type of genotype is it means reference alternate allele is like the reference allele, or it is, then it will say 01. Sorry, 01 is when reference allele. I will explain you the next slide. It will be more clear to you what does each thing indicate. See, if it is 00, GT is 00. Four parameters are there. GT, genotype, AD is allele depth, DP is the depth, uh, total depth, and GQ is the genotype quality, and PL is the genotype likelihood. So what you see that GT, what, how many types of GT information can be there? There can be information 00. If it is, if you get in the VCF file, that means that there is a reference homozygote. Your sample have allele just like the reference. If it is 01, means there is a reference alternative heterozygote. Means there's an alternative form compared to the reference. Reference has C, then your alt is A. So it is an alternative. It doesn't have same as your reference. So it will become, it will be mentioned as 01. If there's an alternative homozygote, means both the alleles have different uh, different base compared to the reference, then it is both have it is a alternative homozygote. And if it is 02012, it is possible only if you have more than one alternative allele present. That is, it is not biallelic, it has higher number of alleles, and this dot dot shows that it is a missing value. Okay, then these all I have explained, that allele depth, total depth, genotype likelihood, and genotype quality. All these are mentioned in your VCF file. What, how much was the higher the allele depth, more you confidence you have, good, higher the total depth, more number of reads are significantly supporting that. If the depth is higher, may different it, the later will include only reads significantly supporting the alleles. Means more the depth, more the reads are supporting it. PL likelihood, genotype likelihood is calculated based on this formula, which is a probability based formula. And genotype quality is also just like a genotype likelihood of that is the just PL, that is genotype likelihood of the second best genotype. So this is the detail which you get in a VCF file. I feel all of you must have run and got the result from RefMap PL. Okay, great. So now the next step is that we have used a reference genome to identify our variants. Now the next step we will be using is a de novo based identification of the variants, the SNPs. So we will run de novo.pl to get this result. See, I have run this de novo.pl since afternoon. It is running because it will take bit time. What is the input? I will explain you. De novo.pl will take the we will give the threads, we will give the output directory, we will give the samples name, and we will also give the, and here the sample will not be the aligned file. Here the difference is that, the biggest difference is that it will be taking you the files, not the alignment file. These are the files which are the raw reads files, which are the trimmed read files. Why? Because we are using de novo assembly. Here we do not give any reference genome, so we are not using the alignment file. We will be giving the files which are the raw files obtained after trimming, and we will be giving the PopMap input file. I will copy this command in the chat. So I've copied this command in the chat. I will also explain you a little bit more about it. What does de novo PL does? De novo underscore it must be. It is de novo map.pl. Sorry for that. It is de novo underscore map.pl. So what input we have given it? Similarly, like the sample directory, which sample directory will contain path of the sample read file, not the alignment file, because here we're not using a reference genome. Then a pop map file, which will contains the detail of the population. The output file, the output directory, we can use the option remove PCR duplicates so that our variant calling is correct. And rest, if you want additional options, then that can be mentioned based on the number of mismatches you want to be allowed between the stacks, number of mismatches to be allowed between stack and between individuals. Vary. This is the var alpha is the significance level at which to call variant site. GT alpha is the significance level at which to call the genotypes. Paired end options are that what if your reads are paired end, remove PCR duplicates. Then in the population filter, you can use the minimum sample per population required, which is when it can give a variant at a location. You can give the P, that is the minimum number of populations at a locus must be present. These are the options which can be added compared to the normal option. These are the essential things which you have to give. If you want to add additional option, you can add. It will take time, but don't be impatient about it, because mine is running since afternoon. See how much it has completed. It has run 3 sample. I started at 1 p.m. and my processor has maximum 20 threads. So it is running because it is basically preparing first a de novo mm, assembly, and then it is aligning our reads again to that assembly, and then identifying the variants. Therefore till now it has run, if you see, till the 3 samples. So once I get the result of this, we will be discussing that in the beginning of the class tomorrow. And what other things we will be discussing is that, see, this is the VCF file, variant file, which we have identified, and I've explained you what the VCF file has. This VCF file is just I have finally generated it for all the 200 individuals of my population. I have shared that VCF with you in the data folder, and that VCF now we will be seeing how to convert that VCF to a format which can be used for association mapping studies. And we will be seeing that how to filter that VCF, because that variant call format file, because the file contains many variants which are not of good quality, which have low allelic depth, which have there are many types of filter which can be performed on it, so that we only take in our further association study those variants which have a good confidence, so that we do not give a false prediction. So in this VCF file, if you use BCF tools, is a very common tool, and you can install it by sudo apt BCF tools. And if you use the stat function on it, it will give, and in the output it will give you the indels and the SNPs present in your VCF. Means it will give you, like, if I have run here, see, I have run BCF tool stat SNP VCF, this command I have run. So it gives me the output file, the VCF stats, which contains this value, that these are the number of samples, because I have run on 4 sample, which I have shared with you. These are the number of SNP I obtained. These are the number of indels I obtained. It gives all the information of the total file, the overall stat of the file. It doesn't give individually for a parent or progeny. It will give you the overall stat of your file, of your VCF file which you have obtained, which will be further converted into other format. And first we will be filtering it. For filtering, we will be using VCF tool only. Filtering, the option is that we will take it filtering based on quality greater than 20, that is the, as we discussed yesterday. And this is the input VCF file, and this will the filtered VCF file. And if we want to annotate it, we can use SNP-EF is one of the software popularly, if you want to annotate using SNP-EF. And but the main step, our aim is to go for Jivas, so we will not going on annotation currently. First we will be using this filtered VCF, and using that filtered VCF, we will be going converting it into the HapMap format so that we can use it for the association study. And I have the phenotype of these samples. Whichever study you are doing, you should have a phenotype of that. I have the phenotype of that sample, so I will be using it to run the association study. So I hope you are clear till now. Yes, great, Bharti, that you are clear. Mine, I'll once check. And anyone has read the papers which I have sent? Any of the paper glimpse, if you want to discuss, we can discuss that.
We just got in the evening, so maybe we can have some questions tomorrow.
Okay, sure. You can this paper is see such a big data set they have used. They used a genome-wide decision study of 1400 cases of 7 common diseases and 3000 shared controls. This is the larger the data size, the more accurate results we get.
I have one question. So this stack we can use on any organism data, or is it specific to some organism or plants?
No, nothing. It is not at all specific. You can read the manual. As I've explained you the diagrams, it was being used on a fish data set that
Yes.
is an animal species. It can be used on any data, but you have to give the correct population map information. The
Okay.
information should be correct. That
Okay.
if you do not provide a correct population file, you will get a wrong result.
Okay.
So you have to be very
Hello, ma'am.
specific. Hello, yeah, please say.
Hello.
Yes, ma'am. Yes, ma'am. I have one question.
Yes, please say.
Ma'am, yes, ma'am. In genotyping by sequencing, we take a part of a segment of a DNA, segment of genome.
We take total DNA as input, and that is restriction digested, and then further many steps of library preparation takes place, and then it is sequenced.
Ma'am, actually, genotyping by sequencing is we generally take a part of segment, part of DNA.
No, see, input is always total DNA. I have clearly explained in my first session. If you go and see that video and the slides I have shared, that first step, how, and the paper also I have shared, that the total input DNA is first fragmented. All short read
Yes.
sequencer first fragment the DNA, and that short stretch is then further library preparation steps takes. Here the fragmentation is restriction enzyme based, and then the further steps of library preparation takes place, because
Okay.
sequencing such a bigger size of genome is not possible.
Ma'am, actually, another question. Ma'am, if we compare depth and coverage, depth mean how many times a particular nucleotide is sequenced, and coverage mean the part of a genome that we covered during sequencing.
Exactly.
Am I right?
Yeah.
Okay. Thank you.
Hello.
Yeah, ma'am, I'm audible.
Yeah, you're audible.
Actually, ma'am, what's the technique you're using all this technique? Because I'm not a background of the biology and nothing. So, this what the calculation did you use for that annotation, whatever you did? So did
Calculation.
whatever you're doing this coding, whatever this, because I'm not a fully background
It
of this.
is all Linux based codes
So yeah,
and
that's
Linux based tools which we are using for our data. And
That's I get it's the data you're using. So this kind of code, yeah, this is code. Did we can use the ARIA Python software we can use for this techniques?
see, like there are Python software for variant identification, every software has its own algorithm and they differ compared to each other. Like the most
True.
popular, there are many, these are Stack and this BCF tools, VCF tools. These are very popular software which have been highly cited. So we are discussing these. There must be, because Python now is so popular, there must be some, there are tools uh, which are used for these, but it can only be used when it is cited well and you get the correct result from it.
Okay.
Hello.
Okay. Yes, yes, ma'am. Thank you.
Yeah, welcome. So I am ending the session now, and I hope you all understood what I have taught, and we'll be meeting tomorrow, the class at 6. So kindly read the papers
Excuse
which
me,
I have sent.
ma'am.
Yeah.
Is it possible to share PPT with us?
No, PPT is my
Okay.
personal, and you can see the recordings. Every slide is there.
Okay. Thank
Sure.
So thanks, ma'am, for a wonderful presentation today. And
Yeah,
hope
thank you
you
so
our
much.
participants are also enjoying learning with your experiences. And we will meet again with the ma'am in day 4 of this Genome-wide Association studies session. So with these words, now I seek permission of ma'am for ending today's session.
Yeah, sure. Thank you. Thank you so much for listening me.